the session Magdalena Szczygieł from the DKFZ German Cancer Research Center, who will tell us about lung cancer progression.
Yeah, hello. Wait, let me just share my screen. Do you see my screen?
Yes, not full screen yet.
Exactly, wait. Okay, I hope now. No, it's okay. Hi, so yeah, thank you to the organizers for giving me a chance. I think this is the usual phrase. Yeah, so my name is Magda Szczygieł. I'm a postdoc at the German Cancer Research Center in Heidelberg, but soon actually moving to industry. I will present to you an almost finished work over the last 6 years about how we can investigate progression of lung cancer using various fancy tools. And I gave it a sort of science, like popular science title, What Trash Collectors Can Teach You About Your Neighbors. And I'll try to persuade you that indeed a scientist can be a trash collector at times. So I wanted to think about the lung as it was a house. So if you have a house, you have several families living there, and some of them can pick up some duties. For example, one person would clean up the staircase, and generally they live sort of in harmony. But then it could occur that, you know, an annoying neighbor will move in. And not only this neighbor will be loud and will love to make many babies, so soon you'll be super annoyed with his kids being everywhere. Moreover, he would produce a lot of trash. And this trash will just, you know, flood the whole house and will totally overwhelm the trash collecting system and the trash that the whole rest of the house will produce. So if this annoying neighbor is cancer, then one of the different trash sorts it could produce are secreted proteins, so-called secretome. So as a trash collector, as a cancer researcher, one of my tasks could be to look at this secreted trash to try to understand better how, what does cancer do, how it communicates with the rest of the neighbors, so the other normal cells in a lung, and also how do they try to respond and adapt, and what do they do. So if the cells are producing the trash, we need a way to analyze it. And normally for the trash, of course, you could look at it, but one of the ways of like analyzing what it is would be to just weigh it, right? So you use the scale to measure what trash is there, how heavy it is. We have a similar way to do it with proteins. It's a very big scale for analyzing very tiny fragments. It's called the mass spectrometer. It was already mentioned in a previous talk, but I will explain to you in a bit more detail how it works. So it's a method to analyze. It could be used, among others, to analyze proteins. However, what's also important is that we want to distinguish what part of the trash came from which, whether it came from the annoying neighbor, from cancer, or from the normal neighbor, from the surrounding cell. And if we have here the neighbor, the cancer as this sort of avocado cell, we can make it green by just expressing certain proteins that will label everything that the cell produces. And we can do similar stuff to the healthy neighbors. And in this way, everything that the cancers will produce will be labeled in one way, and everything that the normal cells will produce will be labeled in another way. And thanks to this, when we then analyze proteins by mass spectrometry, we can distinguish, okay, this part of a trash came from cancer, that part of a trash came from a normal cell. How exactly does it work? What I do is when I do in vitro experiments, I seed cancer cells together with the normal cells of the lung. And then I collect the trash, so the supernatants of the cells, so the whole mixture of proteins which is contained there. And then I digest it with special proteases that are also already mentioned today. So other proteins which have the function of digesting other proteins. And in this way, I end up only with peptides. To be able to measure these peptides, we have to ionize them. So we have to give them the charge, because without a charge, the mass spectrometer cannot read them. This is also a good way to filter out the garbage from in between the actual signal we're interested in. So we sort of inject such mixture to the liquid chromatography, and there is a special tip which will perform ionization, and then such ions, so tiny droplets containing our purified digested peptides with a charge, can enter this really fancy expensive machine called mass spectrometry, and there the spectra could be registered. Afterwards, I can analyze these spectra, and since these proteins were labeled, how I described before, they will be either green or red. Well, they don't really have colors, but they will have different labels. I can, for each protein, establish what is the cell of origin of this protein. So I can know which part of the trash came from cancer and which part of the trash came from the fibroblast. Exactly by having a list, and then list of these peptides and proteins, and then sort of attributing them. Of course, it sounds very straightforward, but it's actually super complicated, and I end up with huge networks of interacting proteins and have to make some sense out of it. Just in very sort of shortcut, what is the main things I learn? I can, for example, monitor how the cancer cells grow. So how changing some conditions of their environment can influence their proliferation. I can also look under which conditions they will transform and they will become much more aggressive, and then I can try to stop it. I can look at how cancer cells send signals to recruit the other cells. For example, how they would invite or scare away certain cellular components of the immune system. And most interesting for me actually is I can look at the therapy response. So I can look whether and how the healthy neighbors can help the cancer cells survive chemotherapy, radiotherapy, and whatever usual way of how we try to kill cancer. I think the main conclusion and the most exciting finding of my talk is that it's surprisingly easy for the normal neighbor to become the bad guy. So the cancer cells have really sneaky ways to influence the other lung cells to start helping them, and therefore creating this favorable niche, so-called the tumor bed, or the tumor niche, how we usually call it. And the normal cells will be in no time helping it, feeding it, protecting from therapies, and all sorts of things. And I'm trying to understand how exactly this interaction happens and how can we stop this process, because cancer cells are really easy, are in principle easy to sort of to tackle, to treat, but since they are very unstable and they produce many babies, as I said at the beginning, always one of these babies will escape whatever therapy we give. But the normal guys are much more stable. They are not transformed. They are not cancerous. So if we somehow treat those, we have much better chance to actually treat the cancer in the end. That's it. Thank you very much. I want to acknowledge myself and all my collaborators in Heidelberg, in the university, in the clinic, and at the DKFZ. And thank you very much. And I'm looking forward to your questions.
Thank you very much, Magda, for the really interesting talk and interesting analogies. I just have a quick question. So do you think so you can you're able to specifically target kind of neighboring cells?
Sorry, can you repeat? There was some I didn't really capture.
So I was just wondering if you can, so do you think it's possible for therapy to then target just the neighboring cells and for the targeting to be specific enough?
Yeah, of course, this is super challenging because the easiest way to target cancer cells and how chemotherapy does it is to just kill everything that's proliferating. That's why in this very old types of chemotherapy, people were losing hair or nails or had problems with digestion, right? Because everything, every cell which is growing was dying. So of course, we have to be much smarter and much more specific to try to target just the neighboring cells, but not impair the whole function of the organ. Thankfully for the lung, similarly to the liver, these are two organs which actually are quite good. I mean, liver is really good at regenerating, lung a bit worse, but also we can sort of challenge it a bit and it will make it through. I mean, our lung capacity is quite high, even if it's a bit lower, we don't notice, right? I mean, many people who smoke, sure, they cannot run marathons anymore, but they are fine, right? So if we damage the lung a little bit on our own by targeting normal cells, we will make it. Of course, we just have to control it really well, and we have to get better. But now with the fancy tools like sequencing of single cells and stuff like that, we can understand this tumor microenvironment better and better. Yes, I think we're slowly getting there to really try to inhibit, maybe not kill the neighbors, but at least inhibit the trash exchange between all of them.
Great, thank you.
Sorry, can I ask a question? I don't know how to raise a hand. Sorry, I couldn't find the button. I feel all that confused. Right, I love the talk. First of all, I love the comparisons to, and big fan of raccoons as well. Because I don't know that much about cancer, but I studied aging a little bit at cellular level, and I was, so would similar methodologies be applied to study aging as well, you think? Because there's this theory about like some cells are getting old and they release trash as well, and that affects their neighbors. Or do you know if something is going on around that? Or
yeah, of course. So so like programmed aging, so like senescence, for example, is really connected to cancer, right? Because first of all, cancers have to, so maybe for everybody, generally cells, when we age, cells sort of switch off a little bit. That's why we die in the end, because as we age, we accumulate different mutations and like some things which are really not good. So then our bodies detect it and then we'll sort of shut down. But cancers overcome this. So they have ways to sort of go beyond this point of senescence, so the sort of sleep mode. But of course, this aging, so accumulating mutations and everything, is of course really connected to cancer. That's why it's much more common that old people get cancer than young people get cancer. So of course, this there is so-called senescence-associated secretome pattern, so like the trash pattern of cells which are sleeping. And this is in some ways quite similar to the trash pattern of cells which are around the tumor. We don't yet know why exactly. Maybe somebody on the audience knows. But I think there are similarities. So clearly the cells which undergo the sleep mode change what they produce, and this somehow seems to help cancer. And we just need to understand better how and why.
Because you have such a lovely method for that. Yeah, thank you.
Thank you.
Thank you, Magda. And also I'd just like to take this opportunity to thank all the speakers and for also keeping to time restrictions. So we actually finished this session even a couple minutes early. So thank you very much again.